what I am going to try to do with this talk is give you a feel for what it's like to work in the extreme environments that we hang out in. And there is a real reason for hanging out in the subsurface, besides the fact that it's fun. And that is that it really represents an extraordinarily different part of the planet than we normally experience. We are surface inhabiting creatures, and we tend to think that that is the dominant condition on this planet. But when you actually start going into caves and mines and looking at organisms that live in the subsurface, you quickly realize that you're really looking at a very, very different part of the biosphere than we normally experience. And in fact, the biodiversity in the subsurface is really staggering. There are a number of reasons for this, having to do with the fact that transport mechanisms are much more difficult in the subsurface. So things can't flow around in air currents and within fluid materials the way that they can on the planet's surface. So really, when we look at the subsurface, we're talking about a number of different habitats. This happens to be one of the most magnificent caves in the world. It's only about an hour and a half drive from my campus. And this cave was kind of a sacrifice cave. It was a party cave where people sort of vandalized it and threw trash in it. And about 40 years ago, a group of cavers, known as a grotto, if you're not familiar with caving terminology, caving clubs in this country are called grottos, and they decided to adopt this cave and essentially clean it up. Well, as the years went on, more exploration occurred, and in 2001, one of my students and several other more experienced cavers actually broke into this passage. And what you're looking at here is called Snowy River. This is a beautiful calcite formation in a big underground river channel. And over the years since 2001, we've explored this, and now there is almost 19 kilometers of this, making it the single largest by orders of magnitude of any cave decoration, any cave mineral decoration in the world. So the subsurface holds wonders that are yet to be explored, and caves are extremely diverse. So no one would have predicted that this feature would actually be in here before people actually went and found it. Of course, my interest, besides the mineralogy of this beautiful structure, really actually centers around something that's much more subtle in this picture, and that is dark streaks and marks on the wall. And they're not as beautiful to the naked eye, but they are the product of microbial interaction with the geochemistry of the cave. And we have isolated something like 180 strains of microorganisms, of which only 3 had been previously known. Now, this is not rare for us. We almost never go into the subsurface where we don't get almost entirely novel strains. And many of them we only know from their genetic fingerprint. We don't have any history of them being cultured, and a lot of them are actually hard to culture. So I think of the subsurface really as kind of a continuum here. The basic background habitat is really the rock fracture habitat. And there are fractures in virtually every kind of rock. So the amount of real estate that is in the subsurface for organisms to inhabit is truly stupefying. And then, of course, we have particularly enriched aspects of subsurface rock material when those are water-storing rocks. And those can be sandstone aquifers that have space within the sandstone grains to essentially act like a sponge. But in many cases, when the aquifers occur in carbonates like limestone or dolomite, they are not very sponge-like, but they are massively fractured. So that habitat has its own specific characteristics, and perhaps a quarter of the aquifers on Earth are composed of these highly fractured rocks. And we certainly know that at least 25% of the world's people get their potable water from aquifers in these highly fractured rock types. So that's another part of this grand subsurface environment. And then, of course, we're gifted by geology, by geological processes, with having windows that we can actually get into the subsurface. There are natural, larger cavities that are formed with a variety of mechanisms, and these are a gift to us by nature, and we can get into those so that we, as humans at our size, can actually physically experience this environment that the microbes that live in this environment are experiencing, but on much smaller spatial scales. Of course, because we're humans and we create things, we have minds that we actually purposefully delve into the subsurface, and I think of those actually as anthropogenic caves, because after you make one, even though humans have been the agency of actually creating that cavity, it takes on a lot of the characteristics of a natural cave, and particularly this is true of abandoned mines. So all of these form a continuum of one sort or another in the subsurface. Well, we often travel into these environments, and what we see are mineral deposits like this, or those black patches on the walls. And when you look at them, you don't realize that these are biological products. They look like minerals. And so they have not captured people's attention really as objects of study from the biology point of view until perhaps the last 20 years, and particularly the last 10. The kind of work that I do, there's not more than about a handful of people in the world that actually study these from the microbiological point of view. Now we're working very hard to produce new students who will carry this work forward. There are extraordinary things about the subsurface. And this is one of my favorite examples. This comes not from my work, but from work of friends and colleagues, John and Susie Pint, who do a lot of their work in Saudi Arabia with the Saudi Geological Survey. And they have focused a lot on what's known as the empty quarter. It's this very dry sand desert, and you can see the background picture illustrates that. But within this landscape, and here you can see Susie peering down into this little hole, there are many, many, many caves. Some of them have tiny little entrances like this. If you can actually focus on here, this is a caver rappelling down into one with a very big entry. And the notable thing is once you actually get down in there, you essentially have transitioned to another planet because it no longer looks like this, it looks like this. And these are residual pockets of biological activity. A lot of it is microbial, but there are also invertebrates in this aqueous environment, and these are diveable pools. So this transition, even when you have big entrances between the surface and the subsurface, is completely dramatic. So it really is like having a whole lot of little planetlets under our feet. And if we were the size of microbes, we could actually drill down right now, right under our feet here. It wouldn't even matter if there weren't big caves. We would have tiny pockets of space within the rock material, and that would be sufficiently commodious to allow us to actually live in there. So subsurface environments, there's some really obvious stuff, right? No sunlight beyond the entrance zone. And this is the entrance drop to Lechuguilla Cave in New Mexico, which is the deepest cave in North America, about 500 meters to the deep point. So photosynthesis, as a primary production mechanism, can only provide secondarily received organic material. So cave nutrient trees are rather truncated. But it is a more benign environment than the surface in many cases because it's high humidity and relatively constant temperatures. Now those temperatures can vary with the depth of the cave or where it is located geographically or with the altitude of the cave. But within a given cave, there's usually not more than a degree or two of variability. So from the point of view of temperature and humidity, these are actually benign environments. They're not extreme environments. However, when we come to good stuff to eat, this is usually fairly extreme. These are very low organic environments. But on the upside, they have a lot of mineral exposure. And so organisms that specialize in getting their energy and nutrients not from organic materials, but energy from inorganic transformations of different materials actually thrive in this environment. There are also some caves that I will show you in a few minutes that are unusually rich in certain things like hydrogen sulfide or carbon monoxide. And these come from geochemical sources. Sometimes they come from deep microbiological sources. But hydrogen sulfide, although it's very poisonous to us if we breathe it, it is a great source of energy for organisms that can actually oxidize it and get their energy from that. One of the really cool things about caves is there's no surface weather. And so what happens in caves stays in caves. And so they're wonderful paleontological and micropaleontological and mineralogical and in fact even archaeological repositories. So caves preserve things extremely well. And we can then see the activities of microorganism communities that perhaps have been inactive for tens or maybe even hundreds of thousands of years, still preserved in the mineralogical textures that are left within caves. So I want to talk just for a tiny bit about the unique nature of what we call speleology, which is the sort of official sciencey name for cave science. And cave science is one of those sciences that's actually place-based by definition, rather than topically based. So it really is a combination of all these different basic sciences wrapped up into one. So any of these things like hydrology or aspects of geophysics or paleontology can be considered a type of speleology within the context of that being the place that you are studying them. Of course, my particular interest is what's known as geomicrobiology, the organisms that live in these rock and mineral environments, and so that's what I focus on. But people focus on all different parts of this and still call themselves speleologists. So just a word about caves if you're not terribly familiar with them. These are sort of the longest, deepest examples, just so you have a feel for how far into the Earth's crust we are actually penetrating with these kinds of environments. The longest limestone cave in the world, which is measured by adding up the measurement of all the passages, and you add it all together, even when the system is very maze-like. So we're not talking about 350 miles in a straight line. We're talking about all of those passages put together, is still currently Mammoth Cave in Kentucky. It's an enormous system. It is of great antiquity. It's had 5 or 6 episodes of cave building over its history, and it's protected from degradation by a thick, impermeable caprock system, which actually prevents erosion from destroying the cave in a very fast situation. All 3 of the longest gypsum caves in the world are in Ukraine. This happens to be the winner right now. And here again, that's not an arid environment, and gypsum is very soluble, so you would think how strange that is. But in this case, there's also a thick caprock there that protects those. Caves can be made as a byproduct of volcanic activity. Rivulets of lava that flow down the flanks of a volcano, freeze out on the outside, and then remain molten on the interior until they drain out and leave tube-like structures, and we call those lava tubes, and they're a kind of cave. The longest one, at least on this planet, is Kazumura in Hawaii, although I think there is one in Utah that is not well explored, that is coming up fast to actually give it some competition for that distance. If we look to a planet like Mars, for example, we see that this is actually not much. So the Martian lava tubes can be hundreds of kilometers in extent, but this is the biggest one here. And then very often caves actually are responding to water table motions in freshwater systems or high and low sea level stands. And so this particular cave in Mexico was created beneath the water table. It was exposed by subsequent geological processes and became an air-filled cave. And then as sea level rose, it has now been inundated with water. So it's had a long and complicated history. How deep? Well, currently this is the record holder. in the former Soviet state of Georgia, a little over 1.4 miles in depth. There are several other caves that are contending to push past this. There's a lot of rivalry in caving groups to actually push caves to their deepest and their longest. But we know that even where we ourselves have delved into the Earth's crust, down to the limits of what we can actually do with our technology, we have many, many microorganisms that are still living at these depths within the rock material. The conditions in these mines in South Africa are very, very difficult. They're very, very hot because they are experiencing the Earth's geothermal gradient, and very difficult conditions to work in, and yet completely infested with indigenous organisms. So what do we really mean by geomicrobiology? It's pretty simple. It's just microorganism interactions with rocks and minerals, whatever that is. So the reason that these little guys are so profound is that they really transform materials. In many cases, they actually dissolve the bedrock. And sometimes that's a passive byproduct of their metabolism, but sometimes they're actually tunneling into bedrock. So they are geological agents because of that. And they manipulate the geochemistry a lot. Microbes are fabulous at pushing the pH all around the place, making something more acidic or more alkaline, drawing down the oxygen environment and making something anoxic. And because they do all this stuff, the byproduct of that is that they're precipitating biominerals that are only there because of microbial activity. Sometimes they do this purposefully. They're getting energy out of these reactions. Sometimes their mere presence is enough to start the process of nucleation of minerals. So they just have to show up, and then they will facilitate that process of mineralization. This is important because up until these last few decades, we had no real idea that they were acting as geological weathering agents in the subsurface. There are major erosional forces in the subsurface. Just like lichens on rock surfaces on the top are major erosional forces. But this is true of microbial organisms all throughout this rock fracture habitat. We now are coming to understand that a lot of our economic minerals are actually byproducts of these organisms' activity. So low temperature hydrothermal ores, for example, gold, copper. A lot of the rare earth elements appear to have been precipitated in the presence of microorganisms. For those of us who are rock and mineral collectors, of course, some of the most unusual minerals in the world occur in caves. Many minerals only have been known from caves. And there are byproducts of all of this exotic chemistry and mineralogy that these organisms conduct. So relatively low temperature enzymes that have extraordinary properties are rife in these organisms. It is sad that more people are not exploiting this biochemical richness. We know that a number of the groups of organisms that produce the antibiotics that we have in our current medicine cabinets actually come from groups of bacteria that are extremely common in the subsurface and in numbers that far outweigh the surface. So really the potential for pharmaceutical agents derived from the subsurface is great. And it's unfortunate that Big Pharma has not latched onto this, but they have not. We come up with many, many organisms that are unknown to science. And I'm not talking about just at the species level. I'm talking about perhaps even at the domain level. And biochemistries, metabolisms that are geared to this highly mineralized environment. And of course, this has implications for the very origins of life and the very early evolution of different kinds of metabolism. So there are those aspects of the kind of chemistries we're seeing. And one of my specialties, of course, is really working on development of life detection missions for other planets. And we are using these kinds of organisms and their unusual properties as a template, really, for what life might be able to do, at least what carbon-based life like ours may be able to do. So as we go off on that theme, It's a really hard thing to do, astrobiology. People used to say astrobiology, which was called exobiology up until about 20 years ago, was a science with no content. Because to this point, we have not discovered life on another planet. But astrobiology has a lot of content, and it comes out of thinking of Earth as a planet rather than just the place we live. Thinking of Earth and Earth's properties as another planet is actually very freeing. So this is what has driven lots of us to work in extreme environments, either in the subsurface like me, or places like Antarctica, or very saline lakes and things of that sort. So we're pushing up against trying to figure out what the limits to life on Earth are in terms of physical and chemical properties, and how those types of metabolism and adaptations can relate back into deep time as we contemplate the relationship to what the conditions on early Earth were like when we think life actually got going. And then, of course, how we can manufacture all of these ideas into some coherent understanding of how we go look for that life on other planets. And the more we look for life on other planets, the better we get at interpreting these kinds of extreme environments and their organisms here. So I want to talk for a minute about extraterrestrial caves, because we are studying caves on Earth for their own sake, but we are also studying them as potential subsurface habitat. And the first time this was actually really suggested was in a paper in 1992 that I published with several colleagues. And I was not yet a cave person. I'd never been in a wild cave at that point. But it occurred to us that the last best place to look for life on Mars would not be in the current Martian regime of extreme cold, extremely low atmospheric pressure, very cold temperatures, all of those things, ionizing radiation getting directly to the surface, but it would be to look in the subsurface. And that actually got us started thinking about how would we get into the subsurface of Earth. So what do we actually know about extraterrestrial caves? Well, if you look notionally over here, from knowledge at the top down to wild speculation at the bottom, this is kind of what we know. We know that there are these volcanic caves on a number of bodies, the Moon and Mars and others. So those kinds of processes geologically that accompany volcanic activity are widespread within different bodies in the solar system. And now we have found many such caves. But if you think about this from first principles, any planet with a surface will develop cracks, both from internal and external factors. Tectonic motions within the body, or if it's a very quiescent body, you will at least get impactors coming in, asteroidal bits and whatnot, and smacking into these bodies. And when you have the potential for fluid of any kind, you can potentially dissolve caves, which is the way limestone and gypsum caves are made. Crustal motions can produce subsurface habitat even in the absence of any kind of dissolution by making tectonic caves. And perhaps there are things that happen on other bodies that we have no counterpart for here. So some of the ones that have occurred to us is every time a comet goes around the sun, it becomes geologically active for a while. And you get a great deal of removal of volatile materials, and when that happens, you could create cavity. Would that cavity be inhabited? Maybe not, but nevertheless, in terms of the geological processes, that would be something that might be going on. Sublimation caves from the Martian polar regions are quite possible, and we know of many examples of those in permafrost on Earth. And even weirder stuff. So I don't know if you guys have been following what we now are beginning to learn about the moon Titan, which circles the planet Saturn, but it is a super cold body. When you look at the images of the landscape, it looks very Earth-like, and yet it's produced by the intersection of surface materials that are organic goo overlying water ice bedrock with liquid alkanes as their hydrology. Okay? Totally different chemistry, totally different temperature regime, and yet the geomorphological features on the surface are remarkably Earth-like. And we even believe that we are seeing dissolved, cave-forming-like processes there. So there may be really exotic ways to make holes in the ground on other planets. Well, some of these ideas are not new. We know that really very early on in the Apollo era, people were looking at lunar images like this, and looking at these open rills, and realizing that we were seeing on the moon processes that we have right here. And this shows a very similar example in Hawaii. When these roof over, and you can see this is kind of freezing out at the top here, when those roof over, if they roof over, they can become lava tubes and caves. If they do not roof over, then they remain as open rills. So the unity, really, of these geological processes is something that is quite a number of decades old in our consciousness. What's the current state? Well, here's some of my favorite images. We now know of at least 185 big cave openings on the moon. That wasn't even confirmed for the very first one until about 2009. So this is brand new stuff. We know of at least 300 examples of these giant openings into the subsurface on Mars. This is a system where there are open rills and closed segments of a lava tube. This is on the flanks of Mount Olympus. And even bodies like Venus, which of course is tremendously hot but very volcanic with very strange volcanic features, we can actually see, and it's hard for you to see back in the audience, but along here there is a lava tube. The arrows are pointing to it. And even the amazingly volcanic active moon Io around the planet Jupiter, which is spewing forth all these tremendously sulfur-rich lavas, is producing lava tubes. So they're common as muck. They're all over the place. So we know of those. Well, they're really big. This is from the high-rise data. And as a caver, I look at this back wall and I want to rappel in there, like right now. It's so tantalizing, and you can't see around corners. At least we can't right now. Although there is a group working on multiply reflective time of flight structure from motion technology, if that makes sense to you, to try to see around corners. And this is the application for that. So these high-rise images are tremendously high resolution, 30 centimeter resolution. This is 100 meters across. And I often give talks to people who probably don't know what 100 meters is, so I found something common that you could actually put in there to give people a feeling for scale. This is an American football field, easily fitting. So we could conceivably have a football game at the bottom of the shaft, should we ever have the opportunity to do so. And we've been searching for features on Earth that we could look at, that we could compare to this. So we looked at, a couple of years ago, 2 summers ago, we looked for this feature. Partly, we actually found it on Google Earth. Isn't Google Earth amazing? It's like, you know, technology's gift to us. Because when we look at this thing, it's very Martian looking. Okay? Now, it's not in volcanic terrain, so there are differences. But we were just trying to get a feel for the scale. And when you actually look across that, this is what it looks like. This is Utah, you know, it's the West, so pickup trucks are always in there in the picture for scale. This is 22 meters in diameter. This is a baby compared to the Martian feature I showed you. This is about 80 to 90 meters deep. Now looking for another analog, working in the Galapagos Islands about a year plus ago. This is a big volcanic collapse feature, which is very similar, more similar in origin. This is about 80 meters diameter, rather. And if you look up at the top, you can kind of see a tiny person. Where's my big stick here? There's a tiny person right there. Right. So to give you some feel for these kinds of structures, they are enormous. In fact, we haven't even found any small ones. It's really quite remarkable. If you have been in tourist caves, most of them are in limestone. But the truth is, when I started to work on caves, one of my biggest surprises is that nature can make holes in just about any kind of rock. And here is a sort of a potpourri. Upper left, this is a lava tube that I've just been discussing, but even much more exotic materials. So for example, these are granite caves in Spain and northern Portugal that we're studying. Quartzite, which you think of as really not very dissolvable, but if you add biology to the mix, it chelates the silica in the quartzite and makes it a gel, and you can dissolve these caves out. Lechuguilla cave that I mentioned before is made in limestone, but it's made in limestone with the action of sulfuric acid to actually chew out the chambers. Even rock salt. This is a thick sequence. There are many of them in the Atacama Desert region of Chile. There are rock salt caves in Israel and Spain and Kenya and lots of different places around the world. Beautiful metamorphic products like marble, beautiful caves in California and elsewhere around the world. You can even do it in gypsum. And if you're not familiar with gypsum as a mineral, it's calcium sulfate. It's probably what the wallboard in this room is made out of, right? So it's a very common mineral. And even ice caves. So these caves that one of our graduate students, Aaron Curtis, is studying are formed around fumaroles on the flanks of Mount Erebus, the very active volcano there. So you can make holes in rock. You can make holes in water ice as rock. So the organisms, to turn back to them, this is Lechuguilla Cave. This is a famous chamber, one portion of a famous chamber called the Chandelier Ballroom. These are all calcium sulfate crystals here. And Lechuguilla Cave is probably about 150 miles by now. We know that there's probably twice that much cave there based on argon tracers, tests that have been done. And this is the system that I first worked in. I had never been in a wild cave. We trained for 3 hours and we went into this system for 5 days, and it nearly killed us. And the whole time I was in there, I just kept thinking, I've been in tough environments before. I just have to live long enough to get out of here, and then I never have to come back. And I got bruised, and I got gypsum in my eye, which caused it to swell shut. And I got a big cut on my leg, and I had a twisted ankle, and I limped out of there, going up many ropes, which I was not particularly skilled at the time. But when I got out, you know how humans are so self-delusional and adaptable. It's one of our great assets. All the pain started to fade, and all I could remember were things like this, that I had been in an environment that was unlike anything I had ever seen, and that it was possibly the closest that I personally would ever get to coming to another planet. And it was a planet right underneath our feet. And so I realized at that point that I could learn to cave safely, not kill myself, and be able to work in these environments. So I was totally hooked at that point. So one of the things that we learned about these environments is how highly partitioned they are. So this is an arid land cave with very subdued fluid moving through it. It has many pools, but it has no rivers. This is in the Chihuahua Desert near Carlsbad, New Mexico. And so the organisms that live in here have a very slow pace of life. Many of them are sort of slowly dissolving the limestone bedrock. That releases chemically reduced iron and manganese. And then other members of the community, all on the scale of centimeters or less, are oxidizing that manganese and iron and getting energy out of it and forming the basis of a food chain. And then there are other organisms that are eating them, and all of that is happening over, spatial scales of that. But there's as many trophic levels, as many layers of handing off food to each other as there would be in the Amazonian rainforest, just at a miniature level. So this extreme isolation of microhabitats. We don't even know actually how to handle this. If we take samples of material from here, and samples of material from here, and maybe samples of material from here. And we actually look at the genetic contents, how many apparent strains we have. Maybe we'll get 10 or 20% overlap. This is nothing. We have tens of thousands, probably maybe even pushing into the hundreds of thousands of strains that we've identified in caves that we don't know how to study because the richness of the biodiversity is so high. So that's my colleague Diana Northup's problem. She's our molecular phylogenist. My interest is which of the ones of all of that biodiversity are actually doing the mineral transformations. And for me to actually study those, I have to grow them. So a large part of my work is zookeeper, trying to figure out how to get these guys into culture. Some of them have been very recalcitrant. This is a scanning electron micrograph of some of my favorite ones visually. We haven't gotten any of these into culture. We think of these as the Death Stars because they're almost 2 microns in diameter. These are the tiny guys, and they're clearly nanobacteria. They are not artifacts, in case you're wondering. And we have found them widely in there and completely failed to be able to get them into culture yet, but I haven't given up. And then these are some of my favorite ones. We call these the Smurfs because they have these hairy bits that look like Smurf hats. And all of these unique morphologies, and many, many more. Some look like chrysanthemums, some look like beads on a string, some look like hairy sausages. The morphology of subsurface microbes is much more variable than in surface environments. So we are trying to work our way forward about how we actually grow these guys to see who is responsible for these mineral deposits. Now we have had some success, and we have grown quite a number of strains that are clearly responsible for the transformation of iron and manganese minerals. And one of the things that we have found that is really important and very hard to deal with in the laboratory situation is that microbiology, classically, you try to isolate individual strains and grow those and poke at them with sticks and test them and all that stuff. Many of these organisms don't do what we're interested in if they grow alone. They only do what they're going to do in consortia. So we have been working with what we call MMC, which is Minimum Microbial Consortium, for any given transformation. So if we are interested in organisms that are producing the mineral todorokite, which is an oxide of manganese, we try to isolate the community that does that. And if we take somebody out of that, no manganese oxidation occurs. So this is an illustration, I think, that shows that a lot of microbes do not do what they do in nature when you actually isolate them. And so this makes them harder to work with. So I'm going to take you through a few of the more spectacular, visually spectacular examples of caves that we've studied, but realize that these extraordinary properties are common to caves in general. So one of my very favorite caves is this one, Cueva de Villaluz in Tabasco in Mexico. We've done 3 National Geographic expeditions here, and we've been studying it since the late 1990s. And this system is tremendously biologically rich, not just microorganisms, but all the way up the food chain. 5 species of bats, cave-adapted fish, zillions of spiders and true bugs and insects of all sorts. And it's all due to the hydrogen sulfide, which is poisonous to us. But the bacteria that can oxidize the hydrogen sulfide are the basis of the food chain. And this cave is bursting with life. This background picture here with all these little tiny black specks, these are cave fish. If you've ever been in a regular cave, you would never see this biodiversity or this number. You would see an occasional cave fish. These are so numerous that the local Soke Indians, as part of Holy Week celebration, actually go in here and harvest them. I can't imagine they taste very good, but you know, it's part of the ritual. So they harvest a bunch of them from this cave and take them out, and who knows how they fix them. I'm not brave enough to have ever tried it, because these guys are acid tolerant and they're soaked in sulfur. This whole system is soaking in sulfur. The limestone itself is being converted to gypsum because of the sulfuric acid. It drips on you, and you have to wash it off. We have to go in here with suits, and we have to go in with breathing gear, and we take extra oxygen bottles. And this is everybody's favorite, especially second graders, snotites, right? These are hyperacidic. These are usually about pH 0 on the bottom, maybe up to about pH 1 or 2 up here. And this is entirely microbial goo. Thiobacilli of various sorts living in here. And this extreme acidity is a combination of the microorganisms oxidizing the hydrogen sulfide and producing sulfuric acid, and the natural tendency of this reduced gas also to abiologically also produce acid. So this cave is quite extraordinary. There are many, many stories I could tell you about this. It's a very difficult cave to work in. It's nearly killed us on many occasions. It not only has hydrogen sulfide, it has a significant amount of carbon monoxide that comes into the system sporadically, CO2, some weird aldehydes. It's really a chemical stew of poisonous gas. But we have learned how to cave in there safely, and it's given us an insight to the interaction of sulfuric acid with limestone in a cave situation, in a shallow enough system that's open that we can actually get into. And that relates to other caves that we have worked in, where that sulfur chemistry has been the same, but has taken place way underground. So this is one of the most famous systems we've worked in. We've done 2 National Geographic expeditions there. And interestingly enough, there's something about these giant crystals that really grabs people, because the first National Geographic special we did, which aired in 2008, is the most watched National Geographic special ever in the history of National Geographic. It's been translated into 40 languages. I mean, everybody knows about these things. And this looks like we are wrapping ourselves up against the cold, but it's just the opposite. We are packed in ice suits, essentially. And we have backpacks that are blowing air across ice cylinders in order to keep this cool enough for us to tolerate it, because this is a highly thermal system. It's based on sulfur. And what attracted us was really a colleague who's a cave mineralogist sent me a few images and said, you know, I think we're seeing some kind of microbial fossils in here. And of course, I could not say no at that point, especially with National Geographic paying the tab, you know. So I said, sure, let's go study it. So here again, it's calcium sulfate, right? It's gypsum. The crystalline form is called selenite. And here's the temperature range of where we're working. 105 you can sort of tolerate as a human. 140 you cannot. And this is saturated humidity because the water has only been drained for mining. So in their natural state, these cavities are entirely filled with fluid, from which the giant crystals have crystallized out. And apart from the giant selenite crystals, there's all these gorgeous iron oxide deposits on the walls that we were very interested in. So we didn't know what we'd find. And it turned out we found a lot. We're still working on writing up the technical description. We have not finished that because we're very, 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 very careful, not figuring we need to rush to press. But these are the microbial fossil images that got me really hooked. All of these filaments in here. We've seen microbial fossils like this elsewhere. So when Paolo Forti sent these to me, I was pretty convinced that we were at least looking at a system where we could look at fossil microorganisms. There are holes in the crystals, and these are really big. Inclusions occur in many different rock and mineral types. Very often they're very small, pinpoint size. But in these particular ones, they're big wonkin holes, and they're filled visibly with fluid, and a lot of them have solid material. And we're like, ooh, we want to get that stuff out of there. So that's what we went after. And the walls themselves, those beautiful orange walls that I showed you, are completely thickly clustered with microorganisms. These are electron micrographs of some of that material. This is at a lower resolution. This is at a high resolution. These guys are highly variable in shape. It's known as pleomorphism, when an organism is variable in shape. These guys look like they're diving in and out of the clay. None of these are strains ever described before. We have closest neighbor identifications of not more than 0.9. So these are really unique organisms. And this is a summary of the results so far. So the biggest crystals are about 500,000 years old. We know a growth rate for them from several different lines of evidence that other workers have done. And so we were able to estimate that where we were able to drill into some of these inclusions from the outside was at a time-depth relationship of about 10 to 50,000 years. And we first, of course, looked at the DNA recovery. And we've been able to identify actually more than this now, more like 60 strains. But the most exciting thing for me, since I want to grow live things, is to actually look and see if anybody's alive in these little time capsules. And it turned out there's a ton of things alive in there. And they were alive, and we were able to get them out and actually get them back into culture. I anticipate spending the rest of my career slowly working on these guys and hopefully passing it off to my students and colleagues. We know from work from virologists from the University of British Columbia who were there with us that there's a lot of viral material there. So that assured us that we were really looking at an indigenous community. You don't get viruses without hosts. And so this is a really unique community growing at very high temperature. The closest relatives to these organisms are things like organisms from volcanic soils in Kamchatka, other caves around the world, including Spain, Australia, Italy. How does that happen? And organisms in unusual chemical circumstances like very high CO2, presence of petroleum, things of that sort. A weird bunch of creatures in there. So that got us thinking about this issue of entombed longevity. We've got a bunch of organisms that appear to have been perfectly happy and remain viable for at least tens of thousands of years in little trapped fluid inclusions. This whole subject has been very controversial because the previous data that's been used to argue that organisms can remain alive and viable for geologically significant periods of time have occurred in things that can be compromised, like ice and salt deposits, which kind of flow over geological time. That is not true of ours. Those crystals are not really subject to alteration as long as we do the work carefully and do not look at contaminants. But these are such unusual strains, they're not likely to be people's skin bugs. So people have been poking around at this issue for a long time. You can see some of these earlier claims that people were looking at organisms that had been trapped for a long time. I think our work and work in several other systems that is ongoing now with other people are showing that, yes, indeed, these organisms seem to be able to hang out for a long time. So these are the big questions for that. Can we find your DNA and RNA in these trapped geological time capsules? Can we find you still alive? And more importantly, if you're still alive, are you still capable of reproducing and making many, many more little green creatures? This is really important because if that's true, then that says that organisms can be temporarily buried and re-exhumed and enter once again into the general geobiological population. This means that what we think about linear descent evolutionarily may have this extra complexity going on. So we know that they're tough. We know that they can kind of hang out in sort of stasis for some period of time stored in geological materials. We know that these organisms are everywhere in these relatively low temperature high temperature to us, but relatively low temperature geological environments. And we know that the planet hasn't been free of biological contamination, so to speak, for billions of years. And so one of the things that occurred to us is, why are some of the closest relatives that we have of some of our cave organisms really cousins of things that grow in these deep sea vents? And the answer that occurs to me, which I don't expect to live long enough to see vindicated, is that many of these organisms are intrinsic to the subsurface, that they get dragged around with the rock materials that get recycled through supercontinental cycles, and that they are a continuous population, that they never see the surface, and that these relationships probably relate to prior ancient proximity of some of these rock masses. This is a radical notion. Who knows whether it will ever be shown to be the case. So I want to tell you one more story, and that is the hunt for blue goo. We didn't actually start hunting for blue goo. We actually were trying to solve a problem for Carl Francis, who was then the curator of the Harvard Mineral Museum. And we happened to be on an entirely unrelated geological field trip. And he said, you know, you work on geomicro stuff, and I've got these copper sulfide samples. And every time we clean them off and we put them back in the cases, within a couple of years, they get all this brown fuzz on them. Do you think it might be critters? And I'm like, no idea. So I took these poor disease samples from Carl's collection, and I took them back to my lab, and we started working on them. And lo and behold, yes, it is a microbial transformation where organisms are actually getting energy out of oxidizing the sulfide part of copper sulfides, and they're pooping out elemental sulfur, which then oxidizes into beautiful minerals. This is the mineral chrysocolla. This is azurite and malachite. These are common copper oxide silicate minerals, and they're being produced by organisms as a byproduct of metabolism. So we started looking all around the place. So that's a cool enough story, but there's more. They take forever to do it. We grew them for 30 months before I even saw any growth. These guys have nowhere to go. They live in environments where nobody is grazing them. They have no predation pressure. They don't have to live fast, and they don't. After 4.5 years, we began to actually see mineral precipitation that was recognizable. And now we've got cultures that, even single individual cultures that have been going for 15 years, and they're still continuing to produce mineral. As a byproduct of this, we discovered a very strange thing, and that is that these are incredibly tough. And we discovered this accidentally because of the way we prepare them for scanning electron microscopy, which is that we dry them in air, and then we dry them in a vacuum oven, and then we put them in a vacuum system and we coat them with gold and palladium, and then we throw them in the SEM and we zap them repeatedly with electron beams. Now you think any respectable organism would go, all right, I'm going to die, you know, really, you can't treat me like that. We got out some samples a couple of years ago to look at some old stuff, because we are writing this up for publication finally. And I looked under the scope, just the optical scope, and I saw brown fuzzy stuff. And I'm like, surely not. And when I looked at them, yes, there are fungi and filamentous actinobacteria punching up through the gold palladium coating. We were very suspicious, and I'm like, must be recontamination. So we took samples, we ran the DNA analysis on them, and sure enough, it's the guys. And now we've done this a bunch of times. We've done it 4 times. They're really not caring about being stuck in those electron beams. So they're super, super, super tough. So the message I want to leave you with is that even though we are on Earth and we are working with biology that we think we know something about, we're really working with what amounts to alien biology from our point of view. Those mesh stocking-looking creatures that you see in the SEMs here, we see this morphology in caves all over the world. We don't know who these are. We've been trying for 20 years to identify them. We can see them here. We can do the genetics. We get a whole long dance card of who's there. We can't match this up to these. We can't grow them yet. And we haven't been able to isolate them in individual, in an individual sense, so we can actually just do a genetic analysis of just those. So these are some of the problems that we face with the current techniques that we have. Genetic analysis is great, but it doesn't allow you to hook up logically with what you're actually seeing. And this means that that kind of logical connection is going to be even more difficult when we try to apply that to life detection missions. So here are the big questions that we're trying to solve. We know that on Earth these organisms and many others can be super tough against all sorts of different conditions. We're pretty sure, based on our work and others, that they can be entombed in geological materials for a long time. They can tolerate all kinds of temperature extremes and chemical extremes, and even electron beams. And we certainly know from other people's work that there are organisms that are tremendously resistant against ionizing radiation and UV. And then trying to connect that to the life detection. What are the kinds of subsurface habitats that exist on other planets? Much evidence shows that there are cavities in rock. And could those provide homes for alien microbes on these other planets? We don't know the answer to that, but it's a plausible thing to suggest. It has been suggested that maybe Earth is not the site of the origin of life, that maybe Mars in its earliest history, when it was a much warmer and wetter planet, could have just as likely, perhaps, been the origin of life site. We know that there are meteorite materials that come and go between our two bodies from impactors. And so putting all these pieces together, we're trying to solve this issue. How can we go look for them? And how can we use these subsurface environments on Earth to do that? So obviously, the work that I have presented a summary of really is the work of many, many people on my team over many years and many different sponsors. I love this poem because it really sums up the way I feel about the subsurface in general. It says, "Touch the earth and listen to the rocks, for they remember. They know and remember all that has come to pass here." And this is a final shot of another place in that chandelier ballroom in Lechuguilla Cave with these beautiful mineral deposits, but all the walls covered with living things. All the walls are covered with living things that are busy continuing to chew away the cave and make even more habitat. And here's a caver for scale down there. Here's a person. Thank you. If anybody has a burning question, I still have a voice. Yeah.
That was wonderfully inspiring. How old are the caves? So I'm thinking that if all these things have diverged, so did bacteria come from the surface and inhabit different caves and then all radiate out from there?
The answer is yes to all possibilities. Yeah, so some of the organisms clearly have affinities with soil actinobacteria or other groups that one could imagine come from a soil-derived pathway.
So I don't have an idea as to how old these caves are.
Caves can be anything from a few tens of thousands of years old or less, all the way to the oldest ones that I know have been really confirmed on this planet, about 280 million years in Australia, which is tectonically rather quiescent. And those age dates come from secondary fill within the caves. So there's really a lot of latitude for time. And the caves are thought of from one perspective. They're just bigger fractures within the overall rock fracture habitat. So the gene flow continuity is within the caves, but also as much as it can be transported through the fracture habitat. So what I truly believe, and also don't expect to live long enough to see vindicated, is that there's a certain small percentage of organisms that actually can tolerate and survive burial and diagenesis of the original sediments. I think that Lechuguilla Cave is showing strains of organisms whose closest relatives are marine organisms that used to be an ocean deposit, which was the mountains around Carlsbad are a big ancient reef of the size of the Great Barrier Reef now. And that oceanic history shows that the environment was right, and the presence of these organisms that have marine relatives as their closest relatives, I think, is very suggestive. And it may be that some subset of organisms, these tougher ones that I'm talking about, can tolerate those geological processes. And if that's only even a tiny fraction of a % of what's actually in a marine microbial composition, that's still a heck of a lot of potential microbial life. And if that's really true, then those are living in this rock mass. They find extra expression once that rock mass begins to fracture, and they're already there as inocula when you actually come along later and make caves. So I think in many cases the caves come along after, long after those organisms are actually placed in the rock. But of course you have hydrological flow paths from the surface. In caves with openings you also have air currents that can bring things in. But one of the things that is a big surprise to us is doing a lot of work on lava tube caves, which are relatively shallow. You would think that you would see a reduced derivative population of microbes in them, and that's not true. They also have a high degree of endemism and uniqueness in the biodiversity, and that was a big surprise to us. Okay. Yeah?
Are you doing ribotypes mostly, or any metagenomics?
Both.
Really,
Yeah. That's not my bailiwick, so I am not highly educated about it, other than I asked Diana and she tells me what we now know about them. And that's enough for me, because then I can design growth media to try to go after the ones that I'm interested in. And I do a lot of enrichment culture because I'm trying to find the ones that are manipulating certain materials. But her work is very, very helpful in terms of guiding me. And she's at the point where she doesn't quite know what to do with this embarrassment of riches of the data. So we are trying all the different kinds of techniques. It's pretty clear that we have not converged on biodiversity in any samples we've ever taken. I think that's pretty clear. Even though, you know, one wall in one of our favorite lava tubes called Four Windows, we call favorite wall, because we've done a lot of work there. We don't have anything like the complete biodiversity of this one wall that's probably the size of that. So we don't quite know what to do about this hyper biodiversity. You know, how do we make sense out of it? We're still struggling with that, yeah.
So you and others are going down to these places and discovering thousands of new microbes.
Yeah, zillions.
Right, and you're expecting them to have interesting new properties for pharmacological purposes.
They do.
Whatever.
Yeah.
Mightn't they not be pathogenic? There's so much.
You know, they eat rocks, and we're just a big goo ball to them. You know, they just really, if I take any of the organisms that I'm interested in and I try to juice them up with extra organics, I can't get them to grow. Well, even some that I can grow. If I give them a richer organic environment, it kills them off. It kills them off. They cannot tolerate high levels of organic nutrients.
So you're pretty confident that it's safe to be...
Oh, yeah.
Okay.
No, you're more of a danger to me than my caves are. So keep your Ebola to yourself, you know. Yeah. There's a very high viral load of organisms,
and again, that's not a problem?
They're not interested in us. You know, hosts and pathogens tend to co-evolve. And my organisms are almost universally adapted to such a different type or a different suite of characteristics in their environment than we provide. That isn't to say that we don't have extremophiles on us. I don't know if you guys saw the great study published sometime last year about belly button microflora. But they discovered a number of extremophiles in the belly buttons of subjects who tended not to wash a lot. So apparently they're around, you know, and if you don't wash your belly button often enough, you're going to have extremophiles. Now, they were harmless extremophiles, you know, they were not interested in eating you in any way or making you sick. So, you know, and we have these huge defenses called our own human ecosystem, microbial ecosystem. I mean, it's pretty clear that there is a revolution in understanding, just getting going in terms of medicine, that we are not individuals, we are ecosystems. We have huge numbers of gut bacteria, but all over us, and they're not just there, I mean, they're interacting communally, and we function as an ecosystem, not an individual creature. And that hopefully will revolutionize medicine, but it also gives us a perspective on something like this. It's like, in order to become ill, you have to become ill by an organism that can somehow overcome your entire ecosystem and that wants something that you have. And my guys are much more interested in copper. They'd be more interested in, you know, eating your copper bracelet than they would be in attacking you. Yeah.
I've heard some incidents of contamination where people have gone into caves and they've brought in all sorts of contamination,
Yeah.
degradation of the caves
Big problem.
and so forth.
It's a huge problem.
Besides debris and garbage, but other things start growing in there
Yeah,
and deteriorating.
this is exactly it. Yeah, inadvertent organic enrichment or vandalism-derived organic enrichment in caves with these unique biofloras. If you enrich it, you're damaging the, you know, natural conditions in which that microflora is existing. And well, not to mention, you know, invertebrates in caves. And so caves are probably the least protected and least appreciated of all classes of wilderness. People still use them as toilets and sinks and trash cans and, you know, all sorts of terrible things. Oil spills or, you know, spills of chemicals or something goes into a cave. It's like, oh, well, out of sight, out of mind, you know. But, you know, the preciousness really of this subsurface wilderness is extreme. Probably caves have more very limited range indigenous species of both invertebrates and vertebrates than anywhere else. You know, many things are restricted to maybe one cave or caves in a small region. So caves and cave protection is a big deal. Yep. Yep. Any more? Yeah.
Do you find that you get more metabolic specialization in these communities, in like these low nutrient environments than you would like in a typical?
I think so. I think that the large proliferation of antibiotic compounds is an expression of that. I think that they are in tough competition for limited energy. So perhaps a lot of these antibiotic compounds are, you know, defense to keep competitors at bay. They, when I first started doing this work, I thought we would find a very restricted biodiversity based on first principles. And we found just the opposite. And so this tendency to elaborate a lot of complicated compounds, I think, is because of this tight gene flow restriction spatially and hot competition for what limited resources there are there. So it's an extraordinary thing, and I think, you know, much thought needs to be given to really the evolutionary implications of this, you know, beyond the pharmaceutical applications and whatnot. It's like, why are these communities so intensely biodiverse, and why are they so chemically talented? They can do amazing things. You know, we have are slowly working on organisms that are busy getting their energy out of oxidizing rare earth elements and chromium. All sorts of weird, unpromising stuff, you know, and so that's an element too. for toxic elements like uranium. A lot of the subsurface organisms are just as happy chemically oxidizing uranium as they are manganese. So some of them are generalists, and some of them appear to only be able to do one metal. So, yeah?
In a lot of your description, you described that a lot of the energy comes from oxidation, so this assumes the presence of oxygen.
I'm sorry, I couldn't hear you.
This assumes the presence of oxygen, yet some organisms will use methane.
No, it doesn't. There are other ways to oxidize things. So there's, nitrate respiration and things like that. So they don't necessarily have to have free oxygen.
No, but bound even.
There are, yes. I mean, there are constraints on the reactions that can produce energy. And, I hate working with anaerobes. I mean, so just because it's really fussy and because this work is so difficult anyway, we're going into these really difficult environments and trying to bring these things back. And so we've done much less work with anaerobes than we have with aerobes. But we have some very unusual organisms that actually do a lot of reduction. We have a lot of iron reducers that are aerobic or microaerophilic, and that's unusual. And we haven't even reported on those yet. I need about 3,000 graduate students to work through all this stuff. I can't afford 3,000 graduate students. I wish I could. But yeah, there are limits to what they can do in this environment. Yeah. Is that it? I've exhausted everybody. Thank you.
Thank you.
Thanks.